Dr. Bergman has a couple of books that are available. The first one, he's got, I don't know how many books you've written. We're not selling your 30 books tonight. Slaughter of the Dissidents. This is an in-depth study. Maybe some of you have seen Ben Stein's movie Expelled. Dr. Bergman has done a much more in-depth study of the kind of harassment that academia puts on their scientists, their teachers that have anything to do with the ID movement or creation science. Just came out in 2008. And then also Persuaded by the Evidence, 2000 book. Both of these books are available on the book table. It's just to the left as you're going out the auditorium. Dr. Jerry Bergman is the author of numerous books and papers in professional journals. We're always impressed with this guy's degrees. The way that I count it, you have 9 earned degrees. Okay, I lose track. The ones that are specific to this event tonight, he holds a BS degree in biology, Wayne State University, 1970, a PhD in human biology, Columbia Pacific University, and then a MS in biomedical science from Medical College of Ohio. He is associate professor at Medical University of Ohio and Northwest State Community College in Archbold, Ohio. He teaches biology, he teaches genetics, anthropology, microbiology. What don't you teach there? He is a fellow of the American Scientific Association and a member of the National Association for the Advancement of Science. We are really very pleased to have Dr. Bergman in the house. Let's give a word of prayer before we ask him up to speak. Almighty God, we thank you for this evening, and we thank you that you have granted us an occasion to be here and to broaden our horizons. We do ask for Dr. Bergman that you would put wisdom in his mind and in his mouth tonight for us, that his words might honor and glorify you and bring us greater clarity in this terrible struggle that is involved between evolution and creation. And we pray your mighty power upon him. We ask it through Christ. Amen. Dr. Bergman.
Okay, they asked me to talk about some of the worst problems with Darwinism, and this is kind of like asking if there's a drop of water in the sea. There's plenty of problems with Darwinism. And I can mention briefly my own background. I had a father who was agnostic, atheist. He never really made it very clear, but agnostic might be the kindest term. And my mother was a Jehovah's Witness, and I followed Jehovah's Witnesses and became very disillusioned with them, which you probably can understand, and became not only disillusioned with the Watchtower, but also with religion. I could mention Jehovah's Witnesses feel all religion is of Satan except their own. And so going to the Kingdom Hall, which is what they call their churches, going to the Kingdom Hall, I heard a long litany of negative things about the churches. So I wasn't very inclined to go to any church when I left the Witnesses. Also going to school, of course, I heard a lot of negative stuff about the churches. A lot of negative stuff about the churches. Later on, much of this I found out was simply not true. There's a lot of myths about church history. A common one is that the churches killed a lot of scientists. Well, there's no evidence that the church ever killed any scientists in all history. And this is all Christian churches, especially the Catholic Church, which is blamed for killing Bruno. They didn't. And Galileo is probably the most famous example, and that's another myth as well. There's a lot more behind the story than we commonly believe. So I drifted into atheism, partially because of my study of Darwinism, but I became disillusioned with atheism. It didn't take too long, partially because I realized that atheists seem to spend all their time criticizing Christians and hardly any of their time doing anything positive. And so therefore it became apparent to me that, well, atheism may be true, but a lot of these people are not really very nice people. One can criticize the Christians, but at least they spend a great deal of time helping other people. And I became especially disillusioned with the atheist movement because so much of what they taught in their books and magazines, I knew from my own study, simply was not true. So then I began to explore the existence of God first, and it became apparent that the main reason people became atheists was because of evolution. Evolution is the engine that drives atheism. So I decided to explore whether or not evolution was true. So I took my biology books that I had used in college and listed all of the proofs that they used for evolution. And one by one, I found out through research in the scientific literature that these proofs were invalid. The first one I selected was vestigial organs. Evolutionists teach there's about 180 organs in humans that have no function. If God created us, why in the world would God create all of these useless organs? Seemed to me like a pretty good argument. Well, as I did more and more research in the medical literature, it became very apparent that all of these useless organs were not useless at all. As a matter of fact, most were very, very useful. And then I went on to the other ideas that evolutionists perpetuate, and I realized that the house of cards crumbled. And then when I realized evolution wasn't true, well, there's only two explanations for how we got here. One, we were created by intelligence. Two, we evolved through accumulation of mutations, copying errors. And when I threw one out, that left the other option. So then I began exploring Christianity and other religions as well, and realized that the most scientifically acceptable and historically acceptable explanation for the existence of humans and the world and all we see around us is Christianity. And so I moved into Christianity. And I wrote this book, Persuaded by the Evidence, which is on the back table, because I realized that I was not alone. Many people took the same path that I did. C.S. Lewis is a good example. Made the same route that I did. In fact, many of the people in my book took the same route that I did. So it became very apparent to me that, well, it should be apparent to you, that evidence is critical. And if we think about what Christ did in his ministry, he healed people, that's true, but he also spent much of his time teaching. And he taught using information, giving the people information, knowledge, understanding. And so now in my life, I stress the importance in the church as well as elsewhere. We have got to give people a foundation before they begin that road to Damascus journey. Some people have a Pauline conversion where they're converted on the road to Damascus. Many of us don't. Many of us, the journey is long. Sometimes it's very, very long. And so this issue of evolution became very important to me. And the first section here, I'll give you what I think is one of the most convincing evidences that evolution is not true, it could not be true, it did not occur, and that is a result of science, not religion. Science, not religion. And this presentation I'm giving now, I've given this a number of places, and a number of people have said, yes, I feel this is the most convincing evidence that I have seen that indeed this is the most convincing evidence for creation and against evolution. My theme here is research proves evolution true. Now don't get upset, just let me explain. Okay? The fact is, though, it's going the wrong way. And let me define evolution. Evolution is from molecules to man, or as I prefer to say, from the goo to you by way of the zoo. That's what evolution is. And that's a good way of remembering it, because often when you read books, and I'm reading a book right now by Jerry Coyne, which he says why evolution is true, and as I read the book, I realize that he has no evidence for this evolution from molecules to man. None. What he talks about in the book is microevolution, the small changes, which creationists prefer to call variation within the Genesis kind. That's really all he's talking about. And actually, it's a pretty good book, because he has chapter after chapter about variations within the Genesis kind. But he never gives any evidence for evolution, which scientists define it as from molecules to man. So the problem is not the survival of the fittest, which is a main part of Darwin's theory. Obviously, the more fit are better able to survive. I mean, to me, the best example is it's like saying rich people have money. Well, of course they do. Of course, if you're less fit, you are going to be less likely to survive. That's part of the definition. The problem that evolution has is not the survival of the fittest, but the problem evolution has is unequivocally the arrival of the fittest. They have never been able to come up with an explanation as to where the fittest come from. Darwin has tried, Lamarck has tried, there's lots of theories, but all of them have failed. The most common one utilized today is mutations, and that, as you will see, fails as well. And that is the primary basis of evolution is macromutations or mutations. And
Basically, mutations produce variations. Get this to work. They produce variations that can be selected. So they change the protein coding DNA, they change the DNA, the blueprint for life, and that affects the phenotype. And that's evolution. Ernst Mayr said ultimately all variation is due to mutation. This is Ernst Mayr here. He is one of the most well-known evolutionists in our century. In fact, he just died a short time ago at age 99. Theodosis Dobzhansky, he was a Russian biologist, and he said mutation is the only source of the raw materials and hence of evolution. And Dobzhansky is, again, one of the most eminent evolutionists that have lived during this century. So we're not picking on minor figures. We're picking on the kingpins of the evolutionary movement. So the question is, do we have beneficial mutations? Are they common? Well, I did a literature search, and I found out that from this database, there were 170,000 articles which talked about mutation. 283 in Medline that talk about mutation. Total was about 500,000. Beneficial mutations, only a total of 186. So I reviewed all of these articles, 186, and I found none that were helpful for evolution, and I'll go through some of these. This is one example. This is one of my favorite examples. This is a beneficial mutation, seedless fruit, but it's not very beneficial for the plant. Can't reproduce. It's beneficial for us because we don't have to worry about spitting out seeds, and therefore can be beneficial for humans. Another example is the Belgian blue cattle muscle mutation. And this turns out to be a gold mine, it seemed, for beef farmers. It produces more muscle, and the meat is more tender, and it's low in fat. So it sounds ideal. And this is classified as a beneficial mutation. But that's what they look like. Obviously, they're pretty muscular cows. But unfortunately, there are side effects. In fact, there are so many that they're really not used, as far as I know, not used much today. And a side effect is reduced fertility. And it is beneficial for farmers and consumers, but the result is information loss. And basically, it has a 11 base deletion in a encoding region. So there are breaks when the muscle develops. And when the muscle develops to a certain point, it stops. Well, the breaks are broken in these cows. As a result, the muscle just keeps right on developing. And finally, other factors slow it down. Another example is in 1791, Seth Wright discovered a male lamb in his flock that had short bent legs. And the advantage, you might wonder what advantage this would be, but the advantage is it, thank you very much. It couldn't jump high fences. Sheep, as you may know, tend to be very independent creatures. They do their own thing and they wander away from the herd. Some animals, like dogs, stick together, but sheep don't. And therefore, a herder, a chief concern is to try to keep track of the sheep, right? And therefore, if they have short legs, you don't need to have high fences. Therefore, you can save a lot of time and money. So, beneficial mutation, well, Darwin thought so. In fact, Darwin talked about this mutation, and that's what they look like. The middle one is the Ancon sheep, right here. See the short legs? And there's a normal sheep. Well, it turns out, and Darwin, by the way, thought, and others thought, that this single mutation produces a whole new breed. They call this a new breed of sheep. And so many people thought we get one mutation, we get a whole new species. Just one mutation. So a number of people thought this proves evolution. Well, it turns out this is actually a deformity which is lethal. It's called achondroplasia. And humans also have the same deformity, and there is a man who happens to be an actor who also is achondroplasiac. Not a very nice disease to end up with. You end up with a lot of health problems. Awful lot of health problems. And there's another example of a mutation. Extra arm, that would certainly be useful. Just think of all you could do if you had an extra arm. Unfortunately, though, this is did not turn out to be very helpful because in this case they had to remove both arms because neither were functional. Another example is sheep, a couple extra legs. Well, that's certainly not beneficial. And that's an example of what we think of more often when we think of a mutation. You can see that this child is horribly deformed. And if you're in the medical field, you realize that when you talk about mutations, you're virtually always talking about something that is negative. Another example, this child, the mutation caused the deformity in the entire lower half of his body. And this is an example of a real beneficial mutation, but I'll try to explain how it is not as helpful as evolutionists like to think. This is a ribosome, which is part of the cell, which is important in converting the DNA code into protein. So it's necessary for the cell to grow and reproduce and repair itself. And this is streptomycin. Streptomycin, an antibiotic, fits right in here, and when it fits in here, it turns the ribosome off. The bacteria cannot produce new protein, can't repair damaged protein, and it dies. Big problem for the bacteria. Well, if you get damage here, and you can see that little damage right there, you get damage there, streptomycin doesn't fit in, therefore this bacteria is resistant to this antibiotic. So it is for the bacteria a beneficial mutation, but it's not evolving anywhere. It's still a bacteria, number one. And number two, it may not shock you, but this guy, when faced with this guy, is at a clear disadvantage. If you send a patient home from the hospital, they get a lot of streptomycin-resistant bacteria in the hospital. When they go home, normal bacteria outcompete them and are killed. Because this bacteria has a handicap because of this damage. The damage can be beneficial when you're in an environment which has a lot of streptomycin. But if you're in a streptomycin-free environment, this is not beneficial. And they call this the fitness cost. And it's well known in bacteriology that most of these beneficial mutations are only beneficial in an antibiotic environment, and they also carry a fitness cost, which means in the wild they are not beneficial. Another example, this is one of my favorite. This right here is the CD4 receptor and the CCR5 receptor, and this right here is an HIV virus. The HIV virus, to get inside the cell, must dock with both of these receptors. When it does so, it can get inside the cell. If this receptor is damaged, which in this case it is, there it is, it's damaged, so it's non-functional because of mutation. The whole thing was not produced. Therefore, it does not move into the membrane of the cell. Therefore, it does not exist in the membrane of the cell. Therefore, HIV cannot bind. And as a result, because HIV doesn't bind, the person with these cells is HIV resistant. So the person is clearly has an advantage in catching, or resisting in this case, AIDS. But this is not a result of evolution. This is a result of damage of something. Something is broken. Okay, and I found, by the way, they were all fit this category pretty much. Now, I'm not saying it's not possible for there to be a beneficial mutation that's beneficial in all situations. But even example of blue eyes, and blue eyes, we're pretty sure now, is a result of a mutation. So if you have blue eyes, you have inherited a mutation that went way, way, way back. It's a result of a mutation. And I guess a lot of us think people with blue eyes are beautiful, right? Blue-eyed blonde, is that what they say? So blue eyes can be beneficial. But on the other hand, depending upon how light your eyes are, you have a clear handicap when you're exposed to bright lights. I have a very hard time with bright lights because I have blue eyes. Because I don't have the protection that I need. The natural color of your iris diaphragm is blue. And people with brown eyes have blue eyes, and on top of that, they have the brown pigment. People who have blue eyes like everybody else, but they lack the brown pigment because there's a mutation that prevents adding the brown pigment on top of the iris diaphragm. And as a result, they take in too much light. As a result, they have a hard time in very bright areas. Now, some people get used to it. They can adjust to it. But as you may expect, people with blue eyes are more apt to develop certain eye diseases because too much light goes into the eye, especially if they spend a lot of time outdoors. Okay, since the vast majority, and this is well agreed upon in the scientific community, if 99.99% of mutations are harmful or lethal or near neutral, okay, and if the number of mutations that separate two species is 2,000, we need 2,000 consecutive beneficial mutations to go from species A, if I don't lose this thing here, to species B without a lethal or harmful mutation shutting down progression. So what does that mean? That means in order to evolve a new species, remember 99.99% of all mutations are harmful. So you get one harmful mutation, you go backwards. So how many mutations is it going to get to evolve a new species? Well, one estimate is about 20 million mutations before you evolve a new species. Now, there are a lot of things you can do to play with numbers, but it's a problem, and evolutionists recognize this. They estimate over 100,000 different proteins are used in humans, and all these are produced by the 23,000 genes that we have. So how many mutations is it going to take to produce these 100,000 different proteins? Well, the number becomes mind-boggling, especially considering for each step forward, we take 999 backwards. Best illustration I've heard of this, it's like making money on a store by losing on every sale, but making up because of high volume. And you have to think about that for a minute. But if you lose money on every sale, high volume is not going to solve your problem. It's not going to solve your problem. Okay, de-evolution. Most mutations we now know are near neutral, and that means they accumulate. And a major cause of aging is the accumulation of near neutral mutations. A near neutral mutation means that one mutation is not going to cause a problem. 4 or 5 is not going to cause a problem. Several hundred is not going to cause a problem. As a result, they accumulate as we get older, and as a result, when we're 60, 70, 80, or 90, we've accumulated so many near neutral mutations that, as a result, we have what we call aging, which means that nothing works as well. You don't see as well, you don't hear as well, you don't walk as well, you don't talk as well, you don't heal as well. Nothing works as well because every structure accumulates mutations. Now, people age, but also we know now that species age. In fact, many people feel that's a major cause of extinction. As the species is around for longer and longer periods of time, the species accumulates more and more mutations. The species ages, and eventually the species go extinct. It dies, just like people accumulate mutations and die. So the result is, in my biochemistry book expresses it probably better than I ever could. It says that each child that is born has slightly more negative, harmful mutations than his or her parents. That means you have more harmful mutations than your parents. Your children will have more than you. Their children will have more than you. So what's going to happen? And this is standard part of biochemistry. It's in very clear black and white in my biochemistry book that we use. So what's going to happen? After a while, the species is going to age, and after a while, it's going to lead to the extinction of the species. Is that happening with us? Yes, it is. And so we are evolving, but we are going the wrong way. We are accumulating genetic damage, and as generation goes by, and generation again, over and over, eventually we're going to become extinct as a species. Now, medicine is very aware of this, and they're fighting very hard to reduce the problem by medical treatments, medical techniques, and so on. So they're very aware of this. But many doctors don't think, well, what does this mean, that we're accumulating mutations? Well, it should be pretty clear. It means what they call mutational meltdown. And eventually, when we have too many mutations, we're going to end up becoming extinct if we're around here long enough. Another major problem is what they call pleiotropy. Pleiotropy is the idea that your entire body is highly interconnected. So interconnected that when one gene becomes damaged, all too often, and I'll just show you a picture. This is a biochemistry chart which shows the interconnectedness of your biochemical system. Now, I only could show a small part of this, sorry, I just didn't have room on the screen. But you can see here, if you block any area here, any one area here, I can get this thing to work here. Maybe I'll try another one in a few minutes. But if you get any area it doesn't work, that's damaged, it's going to affect everything downstream. Think about a circuit for a computer. If you take a pair of scissors and cut a wire on a computer, probably the whole thing is going to fail, because the interconnectedness is enormous. A better example of that, which is more common to you, is that every drug has a side effect. Why? Every drug has, and there is no drug that doesn't have a side effect. Everyone has a side effect. And when you take a drug, you read the sheet they give you with it, and it says, contraindications or side effects or something along that line. And if you read it, it causes headaches and constipation and pain and sinus problems and you go insane and death. It lists all these things. And you wonder how they can sell it. Well, because they know every drug causes side effects. Why does every drug cause side effects? Because most drugs act by interfering with these biochemical pathways. And when you block one pathway, you affect everything downstream. And a good example is sleeping pills help you sleep. They make you tired. And most over-the-counter sleeping pills are antihistamines. Okay? Antihistamines block the histamine biochemical pathway so that you have less histamines, and histamines, besides affecting your wake-sleep cycle, also affect your ability to produce mucus. And so, sleeping pills help you sleep when you use antihistamine type, which most of them are. They help you sleep, but they make your mouth dry. Okay? Because they affect the mucus production. So if you have a cold, you take antihistamines to make your mucous membranes dry. And the side effect is what? It makes you drowsy. Okay, now there are other side effects as well, but those are the most common ones. And that's because pleiotropy shows us, we know now, that the entire biochemical system is interconnected. So if you have a mutation, clearly that produces an advantage, you can almost guarantee it's going to produce problems. Antihistamines, you want to go to sleep, produces a clear advantage. It also produces disadvantages. And the same thing we find with this biochemical pathway. If something is beneficial, if you have a beneficial mutation, it's a high likelihood it's going to cause other negative things, because the whole system is interconnected. It all works together. And that's a major problem with building life by producing mutations. And there you can see is a small part of the chart, which is a lot easier to see because you can see the different fructose and dextrose fructose and beta fructose and 1-kinase and mannose and glucose 6-phosphatase and so on. See lots of examples. Okay, interactions, or interactons, interactome is a whole set. And this is where we now realize that all the proteins, not just the biochemical pathways, but all the proteins are connected. And you can see see this protein right here? This protein is related, it's connected with all of these proteins here and all of these proteins here. You see all these lines that go around from this protein? This is connected with dozens of other proteins. Each one of these is connected with other proteins. Well, this is a better example. This one is connected to many other proteins, which is connected to this one, which is also connected to many others. So we can see the interconnections in the proteome are enormous. And this is another example. This right here is a membrane of a cell, and what you have here are receptors. And there are hundreds of different kinds of receptors on each cell. This is how many medicines work. When you take a medicine, it docks at a receptor and it affects something in the cell. But you can see how connected all of this is. So if this receptor changes, it's going to affect all of these things downstream. This is why Damage typically, almost always, causes a problem, because if this receptor is damaged, all of these things are likewise adversely affected and will not function. Again, illustrating the interconnectedness of the biological system. And this is called the signal transduction pathway. And all of these, you can see how many connections there are. See how many and of course they all end up with carrying information inside the cell, which allows the cell nucleus specifically to respond in some way. And one other thing which is interesting, we used to think that this was the start of a gene and this was the end. In fact, we talk about this today in those words. We say, here's a gene, here's the beginning, and here's the end. Now we realize that multiple and overlapping genes occupy a vast majority of the genome. About 94% of human genes generate more than one product, which means this is a gene, this is a gene, this gene here is inside of this gene overlaps this gene, and this gene is inside of this gene. So if you damage this gene, you could well affect this one, and this one, which creates a level of interconnectivity that makes evolution very difficult, because if you're evolving a new gene, it's going to affect this whole set. So if this gene have a mutation, which may produce a better gene product, but it's going to probably adversely affect lots of other gene products. That's a problem. Alternative splicing, where the genes are spliced up in different ways. And isoforms, I'll go through this quickly. And this is where a gene can be spliced in different ways. We call these isoforms. And one good example is Down syndrome cell adhesion molecule, which you can get 95 alternative spliced exons. In other words, from this one gene, you can get 95 different genes. We used to feel the genes were the boss. Now we realize that the splicing is very important, and the gene is only, well, it's like a computer keyboard. The computer keyboard here does not produce the information. All the information's there, I just have to put it in. Likewise, a gene here does not produce the information. The gene is like a computer keyboard, and what we have to do is select the parts we need. So the genes have a boss above the genes, which we're still trying to figure out. And this is one, by the way, I'll sell some biochemistry and cell biology for you guys. This is one exciting thing about cell biology and biochemistry. We discover something new literally every week, and therefore it doesn't get boring. I taught statistics at the University of Toledo for 7 years. I taught the same thing every semester for 7 years. Pearson product moment correlational coefficient, measures of central tendency, measures of dispersion, mode, median, and standard deviation and average deviation and so on. Never changed. Biology, though, changes every week, and the changes are really exciting. You got 38,000 possible isoforms from one single gene. Extremely complex. And one point, the more complex the system or organism, the more mutations are possible. And the best example is that of the 5,000 genes in humans, a quarter affect the nervous system. Why? Because the nervous system has so many genes that control it. It's like saying, which is it easier to hit? The side of a barn door, side of a barn, let's take, when you're 5 feet away, or a stop sign when you're 5 feet away. Well, obviously, the side of a barn is much easier to hit because there's a lot more space. Same thing with genes that affect the nervous system. If there's lots of genes, the chances are greater you're going to hit one causing damage, and this is why so many genes are important in mental illness and mental deterioration and so on. The nervous system is the most complex system in the body. Amoebas rarely suffer from nervous system diseases. Why? It's very simple. They don't have many nerves. Okay, let's skip a few of these. And aging, we talked about that. Conclusions of this section. Although other mechanisms proposed, at its core, microevolution produces only new information by one mechanism, mutation. Mutations cannot provide a source of genetic variation that is needed for selection. Can't do it. In fact, it does the opposite. We go backward. Okay, the next Probably one of the most impressive supports for creationism is the fossil record, which I will show you. We have billions of fossils. They are not difficult. You can go to fossil shows and pick them up for a quarter. You can go fossil hunting and bring home truckloads of fossils. There are enormous numbers of fossils. We have no shortage at all of fossils. And this is the earliest known fern. And if you compare this, this is dated at 300 million years. Whether it's that old, I'm not going to talk about here, but it's certainly very old. In fact, it may not be that old because look how similar it is to modern day fern. Identical. As far as we can tell, identical. And I have hundreds of examples, but I'll just go through a few. Sumac leaf, compare it with the fossil, and you can see the veins here. Identical. Another example, elm leaf. Again, identical, and the vein structure even shows very vividly. That hasn't even changed. The side here, the rough saw blade type side, you can see that very clearly in both examples. Sycamore leaf. And ginkgo leaf, that's very distinct. And you can tell by looking at the fossil evidence that it's a ginkgo tree. Now these are the earliest. The earliest that we've been able to find in the fossil record are over and over and over identical to those we see today. Ponderosa pine, very distinct. A lot of detail we have preserved. And here's crinoids, or sea lilies. Here's a modern day crinoid, and here we can see our fossil date at 345 million years old, and you can see identical from what we can tell from external morphology. Starfish, again, very distinct, very clear. And do we have any evolutionary precursors of any of these forms of life? None. Herring, and you can see even the swim position is very similar. The location of the fins, very similar. And we have an enormous number of herring fossils. These are very, very common. Many come from the Green River Formation in Wyoming. They produce truckloads of fossils. Trout perch, and here we see some of the internal details, some skeletal details. So when we examine the skeleton here and compare it with this, we see they are identical. Garfish, we can see the very distinct snout, and again that's very distinct in the fossil. Scales, very clear here, very clear here. If you look carefully you can see the pattern, and the pattern's identical here. So even the scale pattern hasn't changed. How about insects, or spiders? Well, here we go. We have good fossil records here, and that's unequivocally a spider. We can see even some of the patterns on the spider, same as those existing today. Millipede, same thing. It's not the best fossil I found, but I guess I need to do some more looking. Dragonflies, very common in the fossil record, very distinct, and identical to a modern day, including the vein structure here, which is very clear in this fossil. And in amber, we have a lot of insects found in amber, and obviously a winged ant. When we compare that to modern day winged ants, identical or close to it. You can tell what that is without even looking at the label. It's a grasshopper, very distinct, strong legs. And there's a bee. And even when we look at internal structures, a technique here called X-ray advancement, we are able to look inside of, in this case, a spider. And you can see the detail here. And the next slide shows you internal structures. And again, what we find is they are identical to those found today. So this technology allows us to look at the inside and notice that they are identical to that found today. And lobster, obviously a lobster. We examine this very carefully. We can see there are virtually no differences whatsoever between the ancient lobster here and a modern day lobster as well. And a turtle, again, the first turtle is a turtle. The first turtle clearly has all the traits of a turtle. One difference, by the way, we do find is that in the fossil record, a lot of animals are bigger than those today. Dragonfly is a good example. Some dragonflies were huge. We don't see those that large at all today. So there is one difference, that the life forms today are often smaller. And a bat, again, you can see the detail. In this case, we have marvelous detail preserved. No question, it's a bat, and we can also identify the species of a bat. Crocodile, you can see the detail, and there's a modern-day crocodile. And here's a lizard. Excellent bone structure is preserved, and if we were to do an X-ray analysis, we'd find the bone structure is identical or very close to it. Snake, you can see a snake here, and over here. I just did an article on snakes, which was published this month in the CRS Quarterly, and there's no evidence whatsoever for snake evolution. It's interesting to read what all the experts have to say. They disagree among themselves. One person says the snake evolved from this guy, and somebody else says, nope, it evolved from this guy, and somebody else says, nope, it evolved from this guy. We have plenty of snakes in the fossil record, but no evidence of snake evolution. Some minor changes, you see. Salamander, same thing. And this is what Darwin predicted we would find. This is the only illustration Darwin had in his book, The Orders and Species. And what we would find, he said, and he was confident we would. He knew we didn't have evidence then, but he said if we keep digging up fossils, we're going to find that these are the original forms of life, and they just, like a tree, so they call this a tree of life. They spread off of here. And from that tree, and I have hundreds of these, I collect these things, they're kind of nice to make comparisons. And this is supposed to be a tree of life, and you can see crocodiles up here, and various reptiles and amphibians and birds, aves and so on, primates. What do we find? Well, I'll only show you a couple of these because, again, there are hundreds I have, but this is a chart of what we've actually found. Sponges here, and moss, brachiopods, mollusks, arthropods, echinoderms, and chordates. So what we find here is chordates appear here in the fossil record, become more common, become less common, become more common, and become much more common today. But you'll notice none of these are connected. This is fact. The trees of life that are shown are not fact. They're guesses. This is drawn from data. When we draw from data, this is what we get. And again, I have hundreds. Now this one, they're trying to show you the connections, and they're showing you that by dotted lines. So they're honest. But this is what we have from data. And this is what we postulate evolution did. But you'll notice none of these dotted lines, none of these are connected. Now there's some here that are connected slightly. But by and large, all we have is lots of dotted lines. What we have is animals appear in the fossil record, and some disappear. Some appear later. Some are still around today. Some appear very quickly and disappear very quickly. But that's what data shows. Another example, here they're trying to show you how they're connected. But notice they have question marks because we don't know. And this is porifera, which is sponges. So you can see we have lots of sponges. They get more common, less common, but no connections. And when we look in more detail at some of these, this is whale evolution, and this, believe it or not, is one of the most well-documented cases of evolution. Whale evolution. Went to a conference once, and the professor of paleontology there was asked to present the most well-supported evolutionary example of in the fossil record, and he chose whales. We can see here this animal slowly changes. The legs become shorter. Well, seem to be jumping from here to here. And they jump from here to here, jumps from here. Well, now these are pretty similar. And then there's a modern day whale. But when you actually look at the animals, they look very different. That is the animal that they're showing you here. This is the animal they're showing you here. This here, showing you here. Here, this is the animal they're showing you here. And here, and then a whale. You can see when you try to argue that this guy evolved into this guy, it's not very credible. So what they have to do is they have to change the relative size of the fossils to make it look more impressive. But on the other hand, if you saw pictures of all these guys, there are animals today that look very, very much like this guy, this guy, this guy, this guy, this guy, and this guy. So all they're doing is taking extinct animals and lining them up and saying, well, this guy evolved into this guy, and so on. And they have fancy names for these, Ambliocetus and Rhodocetus and so on, and this, of course, is the final level. By the way, Darwin thought that the whale evolved from the bear. Many argue that the whale evolved from a dog-like animal. There's about 12 different animals that they've speculated that whales evolved from, none of which are very convincing. And we see evidence of evolution in the fossil record. We can see we have hundreds of fossils. We're able to compare them, and we date them. Okay, and you can see this guy now changed a little bit over here. Whatever they're measuring here, I forget. I guess the ribs here. These ribs right here they're measuring. Got a little bit larger, a little bit smaller, a little bit larger, a little bit smaller, a little bit larger, a little bit, well, about the same, and a little bit larger. What do we see? Back and forth. And these are very minor changes. In fact, the most dramatic one, well, let's see, here to here, almost no change. Here to here, probably this is the greatest amount of change. But again, we're talking about very small changes. And we can see the back and forth is very clearly found. And this is based on data. This is one thing you have to do in looking at evolution is you have to look at data versus speculation. When you look at the data, I rarely ever see any problem. Data shows microevolution. Here you can see there's some evolution here, but we're talking about microns. So they're measuring very, very small changes. And these, I think, are plankton. That's what they are. And evolution's Big Bang. This is one thing that evolution are trying to explain. Life seems to appear very rapidly in an era they call the Cambrian. And that's called the Big Bang or the Cambrian explosion, which evolutionists are trying to explain away. But this is the thing I was actually wanting to look at. New discoveries show that life as we know it began an amazing biological frenzy that changed the planet almost overnight. Well, evolutionists would say geologically overnight is a long time. Okay? But still we're talking about changes, rapid changes that evolutionists have not been able to explain. Okay, the next PowerPoint. This next one deals with what is probably the most controversial area in the entire creation movement. And I find that ironic because I don't see any problem at all with it. Very logical to me. And this is the question, is how long it took God to create. Now, listen carefully. Follow what I'm trying to say, so you understand where I'm coming from. And first of all, the Bible clearly says God is the creator. Scriptures teach God created the heavens and the earth. The word creator is synonymous with God. I'll go through this quickly. We don't have to focus on this. A lot of scriptures say the same thing. Question is Genesis myth, fact, or fable? And no doubt the most controversial books in the Bible are the first and the last. Right? Genesis and Revelation. To me, it reminds me of a plane taking off and landing. That's when problems typically develop, is taking off and landing. Okay, the church is actually split over this. They say the earth, well, I'll give you a couple examples. Is it 6 days old or 14 billion years old? First of all, there are not one or two positions. There's 35 different positions on this one question. So I'll just skip all of them and I'll stress what I've concluded, my position. Number one, it's a miracle. Whether it took 6 days or 6 billion years or 60 billion years, it's a miracle. You have to accept, first of all, it's a miracle. In fact, it's the greatest miracle in the Bible, right? The creation of the earth. But Scripture does say it took 6 days. And there are many books which document that. The only way you can logically interpret Genesis as well as other scriptures is you're talking about 6 24-hour days. Scientists says 14 billion years. So how can you believe in Scripture when modern science says 14 billion years? One example is you've got 100,000 galaxies. Each has about 100,000 stars. How could God create that in 6 days? That's absurd. This is more commonly used to argue for an old universe. The farthest known galaxy is 11.2 billion light years away. That means that when we see it, we are seeing a star that existed 11.2 billion years ago, right? Okay, it takes 11.2 billion light years for light to get here. And by the way, this interpretation is exactly what Henry Morris said in the Genesis flood. In fact, he said it better than I did, so I apologize. He actually did a very good job explaining this in one short paragraph. And here we can see some telescope evaluations. This area right here blown up here, this area here blown up here, and we can see some fuzzy stars which they date way far away. Now here's the main part of the illustration. How old is Adam here? Here's God, here's Adam. When God created Adam, how old was he? He was 0. He was just created. Now if we take our Adam, a couple of you have sons that are doctors. So we'll take our Adam and will bring him to you, your son, and say, okay, how old is Adam? And your son, the doctor, would look at him and say, well, let's see. Physically, he looks about 25. And face here is kind of a teenage face, but his body looks like he's about 25. I'd say he's about 22. So scientifically, he's 22. But how old is he in fact? He's assuming it takes a day to get to his son, the doctor. He's a day old, right? And that's called creation ex nihilo. And everything that is created ex nihilo from nothing, and that's what the scriptures say, everything that's created ex nihilo always has the appearance of age. Let's say God is trying to impress me and I don't have a pencil. I actually do, but God creates a pencil ex nihilo. See, I don't have a pencil. And God says, oh, there's one. I would look at this pencil and I would say, well, God just created it. This pencil is about 30 seconds old, but I look at this and what am I going to say? It looks like it's, well, it had to grow the tree, you had to cut the tree down, you had to process the tree, you had to glue the halves together, you had to put the eraser on there. This pencil would have the appearance of being, well, a year, 2, 3 years old. Can you think of anything that was created ex nihilo that didn't have the appearance of age? Nothing. If God creates a chicken ex nihilo, and here's the chicken, he created it 30 seconds ago. I look at that chicken and I say, well, it grew up and it hatched from an egg, and the egg was laid by another chicken, and I would say that chicken's about, I don't know, how old is an adult chicken? A month old, a year old? I'd say that chicken is a year old. So everything, think about this, everything that's created ex nihilo would have to have the appearance of age. You cannot create anything ex nihilo without the appearance of age. Everything would. So God says he created the universe in 6 days, okay, and it has the appearance of being very old. Well, so does everything that was created ex nihilo. In fact, it is the greatest miracle in the Bible because the universe looks like it's 14 billion years old, although when I was in school it was much younger. When I was in school, the earth was only 2 billion years old, and I remember because I had to memorize that. And now the earth is 4.6, although now it's getting to be a little bit younger, it's 4.5. So it's gone from 2 billion years to 4.5, 4.6, and now going back to 4.5. So all of these dates are what things appear to be. The scriptures clearly tell us, though, how old they actually are. And that's the greatest miracle in the Bible. And let's take a few other examples. Science says for Adam, he's 25. Medical exam says 25. Oh, one thing I should mention that. Certain measures would say he's 25. Other measures would say Adam is not 25. Look at his DNA damage, for example. What are we going to find? Probably there's not any, or very little. So judging by DNA damage, your doctor would say, boy, we did a sequence of his DNA and there's only one mutation. His DNA damage indicates he's really young. How about atherosclerosis? My guess is when Adam was first created, he probably didn't have atherosclerosis. His arteries look probably like a 1-year-old or 2-year-old. So some indications are going to say Adam is young. Other indications are going to say Adam is old. And when we look at the scientific evidence for the age of the earth, there's the creation of Adam and Eve. Same thing with Eve. As soon as she was created, she's not going to look like she's a day old. She'll look like she's 25 or whatever. And of course, the scriptures clearly teach Adam was real. So in Adam all die, so in Christ all will be made alive. And there's 4 or 5 scriptures I mentioned that shows that Adam was clearly spoken of in the scriptures as real. The Romans, Corinthians, Genesis 5:3, Genesis 5:4, 5:5, lots of examples. Okay, so Adam must have been created rapidly. There's no way that Adam could be created slowly, right? He'd have to be created as a fully functional, living human being. He could not be created as a part, even a large part. You'd have to be created as a fully functional human being. Only a complete functioning body is logical, period. Blood must be circulating in his body as soon as he was created. Otherwise, he'd end up with brain damage. So we have the same problem with the universe. Science says it's 14 billion years old. Scriptures say 6 days old. And there's an example. When we look at this method of dating the solar system, 60 million. This method, 10,000. This method, 1 million. This method, 5 million, 83,000. And I have pages of these, by the way. This one, 100 years, well, that couldn't be right. So what do we see from all of this? Some indications say it's old, some indications say it's young. That conforms perfectly with the creation account taught in Genesis. Perfectly. So I don't see what the problem is. And again, if you look in the Genesis flood, which Henry Morris wrote towards the end, he does an excellent job comparing the creation of the universe as a fully functional working unit, which we realize now has to be created that way, to Adam. Any questions?